I am Archana from Chennai branch. I have completed MSc in Biotechnology from Kuvempu University, Shivamogga. I am going to present the seminar topic about nephrology. So, nephrology is the study of kidney along with their disease conditions. So, kidney is one of the important organ which helps in the blood purification and excretion of the waste products. So, also it helps in the water balance and electrolyte balance in our body. It also secretes various other hormones called as erythropoietin, thrombopoietin, renin, etc. So in this seminar, let us discuss about the detailed structure of kidney, nephron and the mechanism of formation of urine with the help of diagram. With the help of the diagram, so let's learn about the structure of kidney. So kidney is a bean-shaped organ. So it is located in the posterior side of the abdomen. So here there are two kidneys. There are two ureters, one urinary bladder and one urethra. So kidney is a organ which helps in the blood purification and urine formation. So the kidney is surrounded by three different layers. The outer layer is called as renal fascia, middle layer is called as renal fat and the innermost layer is called as fibrous capsule. So here the concave shape of the kidney is called as hilum. So in the structure of hilum, there are three different structures will arise here. So the first one is renal artery. So in the renal artery, the blood enters into the kidney. So the second structure is renal vein. So where the blood is taken out from the kidney. So here the blood will move out of the kidney. So that is renal vein. So the third structure is renal pelvis. So it is a funnel shaped structure which connects the ureter. So this is about the hilum. So next moving on to two zones of the kidney. So there are two zones in kidney. One is outer cortex and inner medulla. So in the region of medulla, there is a pyramid like structure. We call it as renal pyramid. So renal pyramid will connect the minor calyx. Minor calyx will join to form a major calyx. Major calyx is connected to the funnel shaped structure. We know it, that is renal pelvis. So renal pelvis will again connected to a long tubular structure that we call it as ureter. And the ureters are connected to the urinary bladder to the urethra. So now if ureter is, so ureter will carry the urine to the urinary bladder. Ureter will carry urine to the urinary bladder. So urinary bladder is a muscular sac-like structure that stores the urine. The function. So when the urinary bladder holds about 200 to 300 ml of urine, so we get urge to urinate. Okay. So the function of urinary bladder is to store the urine. So the next structure in the kidney is urethra. So urethra helps in the process of mixturation. So mixturation means the action of urination. So this is all about the kidney. Nephron is the structural and functional unit of kidney. Structure of nephron consists of renal corpuscle and renal tubule. So you can see in this diagram so that the renal corpuscle is the glomerulus and Bowman's capsule. So these two regions are renal corpuscles. So next the tubular structures of the nephron. So all the tubular structures of the nephron will come under the renal tubule. So renal tubule includes proximal convoluted tubule, Henle's loop and distal convoluted tubule. So these are the tubular structures which are present in the nephron. So now let us learn the function of each one of this. So now the first we can see in this diagram the afferent atriole. So afferent atriole and efferent atriole. So these two are the blood vessels. So afferent atriole will carry blood towards the glomerulus. Afferent atriole carry blood towards the glomerulus. So in the glomerulus region, the purification of blood takes place. So the plasma content of blood is purified here. So not any other blood proteins or any other blood cells are purified. Just the plasma content of the blood is purified here. So and the purified blood will go away from the efferent atriole. This glomerulus is surrounded by a cup-shaped structure. So that cup-shaped structure we call it as Bowman's capsule. So the Bowman's capsule will enclose that glomerulus. So we know that in the glomerulus region, blood purification takes place. So the whatever filter is formed, it will enter the Bowman's capsule. So we call it as primary urine. So next, the Bowman's capsule is connected to a tubular structure. So that one we call it as PCT, proximal convoluted tubule. That PCT is next connected to a long hairpin-like structure. So that one we call it as loop of Henle or Henle's loop. So next again Henle's loop is connected to a further tubular structure called as DCT, that is distal convoluted tubule. Next distal convoluted tubule is connected to a collecting duct. Let's see the mechanism of formation of urine. So first step is glomerular filtration. So in the glomerular filtration, so the blood is filtered. So the amount of filtrate which is formed, we call it as glomerular filtration rate. So and the normal GFR rate is about 125 ml per minute. The second step is tubular reabsorption. So in the tubular reabsorption, so whatever the filtrate is formed, so in that if any necessary substances is present, so it is again reabsorbed by the capillary network. So this process is called as tubular reabsorption. So here it may be water, glucose or amino acid, electrolytes. So it will again reabsorb back to the capillary network. The third step is tubular secretion. So in this step, if any unwanted substances is present in the capillary network, so it will again secrete back into the tubules. So it may be excess potassium ions or hydrogen ions, creatinine, hormones and drugs. So these are again secreted into the tubular structures. So this is about the formation of urine. Finally, whatever filtrate enters the collecting duct, so that is the urine. So it passes through the renal pelvis, then to the ureter, urinary bladder, and by the process of mixturation, so it will excrete out of our body. So this is all about nephrology, and thank you for giving me this opportunity. Thank you, ma'am.